Welcome again. This one is chapter 3, all about nucleic acid, which encompasses what DNA and RNA, very important and very crucial for medical stuff. This chapter is, and it is bulky chapters actually. So we have around 4 parts. So this one is the first part, and the introductory part on nucleic acid. So as you know, DNA and RNA are long linear polymers called nucleic acids, and they are required for the storage and the expression of your genetic information. So our genetic machinery or our genetic information can be stored in the DNA, and this macromolecule consists of a large number of linked nucleotides, sugar linked by phosphate from the common backbone, whereas the base vary among 4 kinds. So the genetic information is stored in the sequence of base along the nucleic acid chain. So the base have an additional special property. Among that, they form specific pairs with one another that are stabilized by hydrogen bond. So these base have an additional special property which can be stabilized by hydrogen bond. Therefore, this hydrogen bond can be used for stabilizing the base pairing among the DNA and RNA strands. So the base pairing results in the formation of double helix, and the helical structure consisting of 2 strands in DNA, and this strand seems what a ladder. So the base pairs provide a mechanism for copying the genetic information in an existing nucleic acid to form a new chain. So DNA and RNA are a depository of the genetic information for prokaryotes, eukaryotes, virus, and the plasmids. As you know, viruses are very small infectious particles consisting of DNA or RNA genome protein required for pathogenesis. You know that this virus lacks a complete structure, but have a complete system for replication, the transcription, as well as translation. Hence, virus must invade other cells and command their DNA and RNA as well as protein synthesizing machinery to reproduce. Both eukaryotic and prokaryotic can be affected by this virus, and the virus can infect bacteria. The virus that infects bacteria, also known as bacteriophage. This is the name given for virus that affect bacteria. And the plasmid are small and circular DNA molecule that can enter bacteria and replicate autonomously outside the host genome. So unlike virus, plasmid are not infectious. They do not convert their host cells into factories developed to plasmid production directly. So if DNA and RNA is the genetic machinery of different organisms, so why we study this DNA? Is it important to study about DNA? Is it very mandatory to know about the DNA? Yes, it is crucial. So why we study DNA? Here is that it can be depicted as follows. So DNA serves as storage and the transmission of information. So knowing the mechanism of transmission of information and the replication of DNA is very crucial. And knowing the chemical structure of the gene allows to understand how gene control the inherited characteristics of living thing, and the gene expression is what enables cell of the same organism to take on so many different size, shape, and function. Finally, alteration of the gene expression or function lead to what a genetic disease that is known as the gene mutation, which is associated with different genetically inherited disease. So knowing the concept of DNA and the nature of DNA is very crucial for this and other purpose. When you count the structure of DNA, it is polymer actually, which is made from nucleotide monomers, and the ribonucleotide polymerized into RNA, and the deoxyribonucleotide polymerized into DNA. This simply means that in case of RNA there is single sugar, whereas in case of DNA there is a double sugar that I mean. These nucleotides in DNA are linked by phosphoester bond in twisted ladder shape or a double helix structure. So this nucleotide can be linked by phosphoester bonds. These phosphodiester bonds are between the phosphate on the 5' carbon of the sugar with 3' of the hydroxyl group. That's the mean. So this rungs of the ladder is always a nitrogenous base, whereas the side of the ladder can be sugar and the phosphate group, also known as DNA backbone, and the hydrogen bond hold the base pairs together with 2 bonds between adenine and the thymine, as well as 3 bonds between cytosine and the guanine. This means that there is a triple bond between cytosine and the guanine, and there is double bond between adenine and the thymine. That means so the 2 strands of the DNA double helix run in opposite direction. They can be oriented in one anti-parallel way. Have a look at this. This is adenine and cytosine. So there is a phosphodiester backbone between these 2 ends, and there is thymine and adenosine. The bond between the 2 is, as we mentioned before, it is double bond, but here it is 3 bonds are there in case of cytosine and guanine. So this one is the structure of double helix DNA, which can be shown in an anti-parallel way. This one is the 3', whereas this one is a 5'. There is always coupling of 3' and the 5' bonds, and this one is mainly found in the eukaryotic cell, but in case of bacteria, these 2 may not be found. So this DNA have minor grooves and the major groove here. So different drug can be intercalate this DNA either in a minor groove or in the major grooves. Therefore, this one is the structure of your DNA. So in both double helix, the 2 chains are coiled around a common axis called axis of symmetry, and the chains are paired in an anti-parallel manner, which means that the 5 end of one strand is paired with the third end of the other end. That's the mean. So in the DNA helix, the hydrophilic dihydroxyphosphate backbone of each chain is on the outside of the molecule, whereas the hydrophobic bases are stacked inside the chain. So the overall structure resembles the twisted ladder as I shown in the above, and the special relationship between the 2 strands creates what? The major or wide groove and the minor or the narrow groove. So this groove provides access for the binding of regulatory protein to their specific recognition sequence along the DNA. For example, different certain anti-cancer drugs such as actinomycin D exert their cytotoxic effects by binding into the narrow groove of the DNA double helix, thus interfering with RNA and DNA synthesis. When it comes to Watson-Crick base pairing rule, these bases are paired together in a specific manner. We say that because of the chemical structure and shape, adenine only paired with thymine and guanine only paired with cytosine. So exactly enough room for only 1 purine and 1 pyrimidine base between the 2 strands of the DNA can be takes place. So according to Watson, always adenine paired with thymine and guanine paired with cytosine. It is not possible to pair adenine with cytosine or thymine with guanine. When you come to Chargaff rule, given the sequence of the base on one chain and the sequence of the base on the complementary chain can be determined here. In any sample of the double helix DNA stranded molecule, the amount of adenine equals to the amount of thymine, and the amount of guanine is equal to the amount of cytosine, and the total number of purine is exactly equal with the total number of pyrimidine. Here, this Chargaff rule only focusing on the amounts. So the amount of adenine is exactly equal with thymine, and the guanine is exactly with cytosine. This means that the amount of purine is exactly equal to with pyrimidine. For example, if somebody asks you that given a DNA strand having 30% of adenine, how many percent of thymine according to Chargaff rule? The answer must be exactly 30% of thymine, because of adenine is exactly equal with thymine. Have a look at the property of DNA. This DNA is double strand DNA can exist at least 6 forms, A to E and Z. The B form is usually stable and found under physiological condition, low salt, high degree of hydration. This one is the property of B form of DNA, and it is this right-handed double helix that was identified and hence called Watson-Crick DNA. So single turn of B DNA about the long axis of the molecule contain 10 base pair. The distance spanned by one turn of bDNA is 3.4 nanometer or 34 angstrom, that means, and the width or the helical diameter of the double helix in beta DNA and bDNA is almost 20 angstrom or 2 nanometer. So this one is a diagrammatic representation of bDNA, aDNA, and zDNA. aDNA formed between when bDNA is chemically treated. It is also the right hand helix, but there are 11 base pair here, and the zDNA left hand helix with 12 base pair are there. So it may play a role in the regulation of gene expression. This zDNA can be play a role of regulation of gene expression. Here also the concept of denaturation, the renaturation of DNA. During DNA replication and other processes, the two strands of double helix must be separated from one another, at least in the local region. The two strands of the double helix separate when hydrogen bond and the hydrophilic interaction between the pair based are disrupted. So in a laboratory, disruption can occur if the pH of the DNA solution is altered so that the nucleotide base ionize and disrupts base pairing if the solution is heated. Therefore, if DNA can be heated in a pH under different pH, the DNA structure may be altered. So phosphodiester bonds are not actually broken by such treatment, so DNA strands stay intact but they separate. So the process of changing double strand DNA to single strand DNA is called melting or denaturation or strand separation. So denaturation is simply mean that the process of converting double strand DNA to single strand DNA. The melting temperature is defined as the temperature at which half of the helical structure is lost. This means DNA rich in GC pair melt at higher temperature than the DNA rich at 80. This means that the bond stability of GC is triple bond and the 80 is single bond, double bond, we say that. So denaturation can be monitored by measuring its absorbance at 260 nanometers, since single strand DNA has higher relative absorbance at these wavelengths than those of double strand DNA. So by using UV vis spectrophotometer at 260 nanometers, you have to know what the denaturation of DNA. Inside the cell, the double helix is not melted by heating or pH change. So instead, protein called helicase used chemical energy from ATP to disrupt the structure. Be sure here, or you have to know, identify this concept here. Inside the cell, the double helix is not melted. Therefore, it needs what? The protein called helicase, which can be used for disrupting the structure. Separated complementary strands reassociate to form a double helix when temperature and the pH reach the range in which most organisms live. This renaturation process is called annealing. So this process is known as annealing. So this one is a diagrammatically representation of UV vis spectroscopy, absorption spectra of double strand DNA and the single strand DNA, and the melting curve for DNA. This one is what shows. So single strand DNA have a large absorption maxima than double strand DNA. That means circular and supercoiled DNA. So in some organisms such as bacteria, bacteriophage, and many DNA containing animal, viruses, as well as organelles such as mitochondria and plasmid, the end of DNA molecule are covalently joined to create a closed circle with no free ends. So this retaining the polarity of the molecule, but it eliminates all free 3OH and 5 phosphorial groups. With few exceptions, circular DNA molecule assumes the form of supercoils useful for DNA storage. So you know that our DNA is very large molecules which can be stored in the cell by using what? Supercoiling principle. Supercoils or coiled coils are introduced when the closed circle is twisted around its own axis, or when a linear piece of double helix DNA whose end are fixed is twisted together. So this DNA is very large, so it can be keep in our body with small places by this technique of supercoiling is known as. So supercoil can be a way of compacting DNA to fit a small space or they could also be produced as a result of the process such as replication, and the enzymes that insert or remove supercoil is called topoisomerase. This yellow color indicate that how topoisomerase can be used for what? Cut the strand of DNA for supercoiling purpose. The purpose of supercoiling is, since the DNA is very large, it is very large to have space in our body, or to occupy a small space in our body, we have to what? Make supercoiling DNA. So the next is packaging of DNA. So our DNA is a pretty long molecule, as we mentioned before. Each cell has about 1.8 meter of it. So how does all this fit into the cell? So DNA is tightly packaged into chromosomes, and the DNA is a wound around a protein called histone protein. So this histone protein can be used for what? Tightening or wounding this DNA in small places. So eukaryotic DNA binds to nearly equal weight of the histone, which are small basic protein containing a large amount of arginine and lysine amino acid. Thus histone are positively charged to bind the negatively charged DNA molecule by ionic bonds. So the complex of DNA stranded DNA, histone protein, non-histone protein, and a small amount of RNA is called chromatins. So chromatin is complex of double-strand DNA having got histone protein, non-histone protein, and a small amount of RNA. There are two varieties of chromatin actually, distinguished with how intensely each form is stained. They are EU chromatin and heterochromatin. EU is true, which means that stained light is less intense than the heterochromatin, is transcriptionally active, and the heterochromatin is densely packed. Dark stained chromatin is only found in eukaryote and is transcriptionally inactive. Therefore, in case of euchromatin, it is transcriptionally active and less intense, and heterochromatin is what's transcriptionally inactive and it is dark stained. The result of facultative heterochromatin is heterochromatin DNA that becomes active during a specific developmental phase and that becomes inactive elsewhere. So this one is known as facultative heterochromatin, which is active during a specific developmental phase and then become inactive somewhere or elsewhere. So the histone protein includes protein or enzyme involved in DNA replication, repair, regulation, transcription, processing, and transport to the cytoplasm. The function of this histone protein is to condense and package DNA. Several gene regulatory roles and DNA repair roles also are there. So the histone package and order the DNA into a dense spherical structure unit called what? Nucleosome. And a single nucleosome may contain what? One repeat unit, which having what? 200 packaging protein plus associated proteins. So this one is a diagram of histone protein, how this histone can be formed what? Octamers, meaning having what? DNA in octamer forms to occupy what? Very, very small spaces. So a series of nucleosomes joined by linker DNA pieces give the chromatin as beds or steering appearance, and the bed of each nucleosome contains 8 histone molecule called an octamer, 2 copies with H2A, H2B, H3, and H4, and the linker stretch of DNA converted by histone or histone 1 connects between what? The nucleosome cores. So wrapping of DNA around the nucleosome core compacts the DNA rings about 7-fold, and the overall compaction in the chromosome, however, is greater than 10,000-fold. There should be higher order of structural organization. So the disc-like nucleosome structure has 10 nanometer diameter and almost height of 5 nanometer. So in the chromosome, the DNA is so densely packed that the smallest human chromosome contains more than 50 million base pairs. So this one is the principle how the DNA can be packaged by using what? A nucleosome, and supported by different histone protein. So eukaryotic genome and the DNA sequence. So some region of the DNA contain useful genetic information which that genetic information is known as coding DNA, and the other region do not. So although bacteria have relatively little non-coding DNA, eukaryotes have a majority of their DNA. In prokaryotic, almost all non-coding DNA is found between the gene of intergenic DNA, but in eukaryotic, not only is non-coding DNA scattered between the gene, but the actual genes themselves are often interrupted with non-coding DNA. So these intervening sequences are known as intron, whereas the region of DNA that contain coding information is known as exon. So most eukaryotic gene consists of exon alternating with intron. This picture, the DNA or intergenic DNA or non-coding DNA having what? Intron, exon, intron, exon, intron, exon, alternative segment. So the intron or the exon segment of the gene that codes for protein and that is still present in the messenger RNA after processing is complete, and the introgenic DNA, which means that the region of non-coding DNA that lays between the gene, as the name inter indicates that lays between. So intron non-coding genes or non-coding DNA region that interrupts the coding region of the DNA or the genes, and it does not code for protein, but it is transcribed and forms what? The part of the primary transcripts or transcription. Structure and the types of RNA. So RNA is a molecule that involved in the transcription and the translation of the information that is present in the DNA. So in some RNA, in some case, RNA itself can serve as a genetic material. Example, in some viruses, also sharing many information with DNA, and the RNA poses several specific differences. So the main difference between RNA and the DNA is this one. For example, if you take DNA is usually a genetic material found mainly in the chromatin of the cell nucleus, but genetic material for some virus only in case of RNA, and RNA means that 90% of the RNA is found in cytoplasm, only 10% can be found in nucleus. And this DNA never present in free state in cytoplasm, whereas this RNA may be present in free state in cytoplasm. When you come to Chargaff rule, works to DNA, but Chargaff rule does not apply for RNA because of here maybe the amount of thiamine is quite different with the amount of what RNA, the amount of what thiamine. So the same is true that when you come to the base, thiamine is only found in DNA, but in case of RNA, thiamine can be substituted by uracil, and the molecular weight of DNA is very large or high weight, almost 3 to 4 million base. When you come to RNA, it is a low molecular weight, around 12,000 base are there. So as the name indicates, messenger RNA or mRNA is a copy of genetic material present in DNA and function as a messenger conveying the information in the gene to the protein synthesizing machinery. So it is carrying the information that saying that synthesis of protein is required. So in mammalians, nuclei, the immediate product of gene transcription or primary transcript heterogeneous nuclear RNA and are very heterogeneous and can be greater than 10 to 15-fold longer than mature messenger RNA molecules. So these messenger RNA molecules are processed to generate a mature messenger RNA, which then enter the cytoplasm to serve as a template for protein synthesis. In eukaryotic, primary transcript has extensive and post-transcriptional modification can take place. For example, it can be removed intron and polyadenine tail at the end of 3' used to mark it for the transcription to the cytoplasm, and guanosine cap at it is 5' for the initiation of the translation. So this one is a phenomena can be takes place by messenger RNA. Transfer RNA or tRNA serve as the adapter for translation of the information in the sequence of the nucleotide of the messenger RNA into specific sequence of amino acid. It is the smallest type of RNA, actually 74 to 95 nucleotide only can be found in this transfer RNA, and like mature messenger RNA, transfer RNA are also generated by nuclear processing of the precursor molecules. So at the 3' of AC mean adenosine, cytosine, amino acid acceptor indicates the specific amino acid to be transferred into the ribosome for the protein synthesis. So there is at least 20 species of transfer RNA molecule in every cell, and at least one to each of the 20 amino acids. So transfer RNA molecule contain not only the usual nucleotide but also their derivatives that are produced by post-transcriptional modification, ribothioamidine, dihydroxyuridine, and pseudouridine, which can be in Latin terms SASPI. All transfer RNA molecule are folded to form 4 loops or arms, and the D loop contain usually a base, dihydroxyuridine. It is involved in the recognition of a given transfer RNA by this amino acyl transfer cyanidase enzyme. So anticodal loop recognize the code for an amino acid or the messenger RNA. Therefore, thiamine, pseudouridine, and the cytosine loop contain unusual uracil nucleotide known as pseudouridine, which can be designated as psi. So extra loop varies in size from one transfer RNA to the other. So this one is how transfer RNA can be used for building a template for the protein synthesis. Third one is ribosomal RNA or rRNA, which is found in association with several proteins as a component of the ribosome. And this ribosomal RNA constitutes 80% of the total RNA content in a cell, and different types of ribosome are found in prokaryotic and in the cytoplasm as well as the mitochondria of eukaryotes. So they are classified according to their sedimentation coefficient during ultracentrifugation using a salt sodium chloride solution with a unit of Seidberg unit S. This one is how sedimentation coefficient of eukaryotic and prokaryotic DNA can be converted. In case of prokaryotic, ribosome are different. So in case of prokaryotic, 17 sedimentation coefficient or S can be converted to two subunits, 15S and 13S. In case of eukaryotic, this ribosome 80 sedimentation coefficient can be converted like this, and this in general show how the ribosome can be different in prokaryotics and eukaryotics. So mitochondrial ribosome are similar to prokaryotic ribosome, but they are smaller, which having 55S rather than 17 sedimentation coefficients. Example comparison of prokaryotic and eukaryotic ribosomes. The fourth class of RNA is small RNA. A large number of small RNA species are found in eukaryotic cell actually, so most of them are complexed with protein to form ribonucleoprotein and are distributed in the nucleus, the cytoplasm, as well as both the nucleus and the cytoplasm. So small molecular RNA or snRNA are the subset of small RNA involved in messenger RNA, ribosomal RNA, and the regulation of the gene expression. Mainly, these small RNA can be used for regulation of gene expression. So small interfering RNA also there cause inhibition of the gene expression by decreasing specific protein production. So this small interfering RNA involved in alternative splicing of intron from exon, and the small cytoplasmic RNA involved in the selection of protein for exporting. So this one is a summary of different class of RNA. So messenger RNA carries information specifying amino acid sequence of protein from DNA to ribosomes, and the transfer RNA serves as adapter molecule in the protein synthesis, which translates messenger RNA code into amino acid. The ribosomal RNA plays a catalytic or ribosome roles and structural roles in ribosomes, and the primary transcript and the small nuclear RNA also have their own role like this. So you have to, this one is summary, so you have to read it. So the next new topic is DNA replication or DNA synthesis. Before the structure of DNA was known, scientists wondered at the ability of an organism to create faithful copies of themselves, and later at the ability of the cell to produce many identical copies of the large and the complex macromolecules, and this is due to a DNA replication process. So during duplication of the entire genome prior to cell division, always before cell division there is duplication of the entire genome are there. Whenever an organism reproduces, the DNA must be replicated so that the descendant or the offspring may receive the complete copies of genetic information. Similarly, when a cell divides, the genome must be replicated, so each new cell gets what complete set of the gene. So that's why different species can be produce the offspring which is similar with the parent, due to this genetic or DNA replication or DNA synthesis. So this DNA replication is extremely accurate in order to preserve the integrity of the genome in a successive generation, and the replication rate in eukaryote is slower in resulting the higher fidelity or accuracy or replication. In eukaryotic, replication only occurs during the S phase of the cell cycle. Actually, we have different phase of cell cycle, but replication in eukaryotic can takes place only in S phase of cell cycle. So the fundamental property of DNA replication process and the mechanism used by the enzyme are essentially identical in all species, and the replication is governed by base pairing rule, or Watson-Crick base pairing rule. So each of the two strands of DNA can serve as a template for the synthesis of new complementary strand producing two daughter DNA. So the semi-conservative nature of DNA replication proposed by Watson and Crick was confirmed by Wilson and the Sahal experiment using two isotopes of nitrogen. So for the first time, this DNA replication can be identified by Sahal and Wilson experiment by using what? The two isotopes of nitrogen. This scientists can propose three possible models were proposed for DNA replication by Wilson and Sahal. The first one is conservative model, which states that both strands of one copy would be entirely old DNA, while the other copy would have both strands of the new DNA. So this one is the law of conservation of DNA, which states that the new DNA cannot be the new DNA that can be created, that can be replicated or synthesis from the parent can be similar with that of the offspring. It means dispersive model was states that when double-strand DNA might fragment, replicates to double-strand DNA, and then the reassembly creating the mosaic or a mixture of all in the new double-strand DNA region in each new chromosome. So here, what dispersive model states that first the double-strand DNA may become what fragment, and this fragment can be replicated and then reassembled to creating what the mosaic mixture of the old and the new DNA or double-strand DNA region for the new offspring. That means the last one is semi-conservative model. This model states that the DNA strand separate and that the complementary strand is synthesized for each, so that the spleen chromatid have one old and one new strands. Actually, this dispersive and the semi-conservative slightly similar, but the process or the way can be different. This is a diagrammatic representation of the three models: semi-conservative model, conservative model, and dispersive model. So you have to see the picture and understand what it means. So in case of conservative, the new DNA or the new chromosome of the offspring is exactly similar with that of the parent DNA, because it is almost the law of conservation of energy. You know that states that energy neither created nor destroyed. So only transformed are there. So here also the DNA can be transformed from parents to the offspring. So this DNA replication can be catalyzed by an enzyme. That enzyme is known as DNA polymerase. And the DNA polymerase use only single-strand DNA as a template. Then after it can be used for what replicates and produce what double-strand DNA. So in prokaryotic organism, DNA replication begins at a single unique nucleotide sequence. That unique nucleotide sequence site is called origin of replication or ORIC. But in eukaryotic, replication begins at a multiple site along the DNA double helix, and the parent molecules unwind, and two new daughter strands are built based on base pairing rule. Therefore, this is all about the introduction part of nucleic acid or DNA and RNA. So next I will prepare the consecutive YouTube tutorial for you. So you have to listen attentively and try to understand this concept, because it is very, very crucial for you guys. So it is very important to give attention and understanding the concept of this DNA, because many drugs can be perform their activity or their function or their pharmacological effects on this genetic machinery. So it is very nice to give attention, and thank you for watching. listening attentively. Be careful to share this video for your friends. So share this video for your friends to get what the understanding of these concepts, and subscribe the channel to get what the consecutive YouTube tutorial for you. So don't forget to subscribe, and you have to put your comment on the comment section of the channel so as to modify something for you according to your interest. So you have to put your comment, so don't hesitate to put your comment. Okay, it is the free channel for you, so please use it properly, and write your comments there, and don't forget to like and subscribe the channel and share for your fellow friends so as to benefit from the channel. So thank you very much.